Hello everyone, welcomes to YouTube video on signal transduction pathways. In this video, I am going to discuss about basics of signaling molecules and types of signaling molecules and signal transduction pathways seen in prokaryotes and eukaryotic system. Before going to know about signal transduction pathways, let us start with signal. Signals are the physical, chemical or biological substances or factors which elicits response in the living system, and transduction is the cascades or series of biochemical reaction in response to a signal that is known as transduction. So let us start with definitions of signaling process. The biological systems are dynamic. That means it always changes with respect to physical, chemical and environmental signals. When different physical, chemical or environmental signals strike to the biological system, the system performs various degree of responses in a series of processes or cascades of chemical reactions known as signal transduction process. For the growth and development of organisms, various internal signals are required, and these internal signaling agents typically brings about their effect by means of a series of biochemical reaction called signal transduction pathways. And these signal transduction pathways seen in both prokaryotes and eukaryotic organisms, that is plants and animals. The general feature of signal transduction is the signal interact with a receptor, and the activated receptor interact with the cellular machinery, producing a second signal that changes the metabolic activity of target cell, and this transduction event terminated when the cell returns to its pre-stimulus states. Then the types of signaling molecules. Signaling molecules can be classified into three broad categories. One is endocrine. The signaling molecules act on target cell distinctly from their site of synthesis. For examples are the hormones. And another is paracrine. These signaling molecules released by the cell affect target cell only in close proximity. Examples of the paracrine signaling is the action of neurotransmitter. Then the autocrine. The signaling molecule produces an effect on the same cell that produced the immunosystem to foreign antigens. Basically, and basing upon the ability to move across the plasma membrane, signals are of two different types. One is called lipophilic signals, another is called hydrophilic signals. Lipophilic signals are lipid-loving signals, and the lipophilic signal diffuse radially across the plasma membrane and active several transcriptional factors which regulate the gene expression. For example, is vitamin A, D, E, K and growth factors. And hydrophilic signals are water-soluble signals unable to enter into the cell and that binds with the specific receptor of the plasma membrane, and it activates the formation of several intracellular hormonal mediators which stimulates variety of responses. And most of these examples of secondary messengers are cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, calcium, etc., which are elicits different responses in the living system. So let us see signal transduction process seen in prokaryotes. And prokaryotic organisms, that is bacteria, receive the signal from the surrounding medium such as pH, osmotic strength, availability of food, oxygen and light and toxic chemical substances, etc. This signal elicit responses such as motion towards light, away from the toxic substances, etc. Bacteria sense and responds to this environmental signal by two-component regulatory system. And this, first of all, bacteria sense and detect the signal by a small family of receptor protein which is present in the plasma membrane, and this signal binds with the receptor the receptor undergo conformational changes and initiate the signaling pathway, and that leads to the different types of response. And the two-component regulatory system consisting of two proteins, one is called sensor protein, another is response regulator protein. The function of the sensor is to sense the signal and pass the signal to the response regulator, which brings about the cellular response and gene expressions. And the sensor protein have two domains, it consisting of input domain and transmitter domain, whereas the response regulator have two domains, one is called receiver domain and output domain. The sensor protein have two domains, one is input domain, which receives the environmental signal and transmit the signal to the transmitter domain, which transmit the signal to the response regulator. And the response regulator also have two domains, one is called receiver domain, which receives the signal from the transmitter domain and activate the output domain, which is DNA binding domain, which brings about the response and gene expression. The signal is passed from transmitter domain to the receiver domain by protein phosphorylation. Various types of signaling seen in the bacteria like osmoregulation, chemotaxis. And let us see the osmoregulation signaling in the bacteria. For osmoregulation in bacteria, two types of porine proteins are responsible or seen in the plasma membrane. One is called OMPF, another is OMPC. The pore made by the OMPF is larger than the pore made by the OMPC. When bacteria subjected to high osmolarity in the medium, it synthesizes more OMPC than OMPF. That's why pore size decreases, resulting less water enter into the cell. But when it's subjected to low osmolarity of this medium, then more OMPF was synthesized, then the pore size increases and more water can enter into the cell. And the sensor protein of osmoregulation, that is known as ENVZ, is located in the plasma membrane, which consisting of one input domain and one transmitter domain. When osmolarity increases or decreases, the input domain undergoes conformational changes and the transmitter domain autophosphorylates its own histidine residue, then phosphate is transferred to the aspartate residue of receiver domain of response regulator OMPR. Then OMPR has a DNA binding domain which binds with the particular promoter region of this DNA, which regulates the gene expression and expresses the porine gene, that is OMPF and OMPC. If it will bind with OMPF, then OMPF protein will be produced, and if it will bind to the OMPC, then OMPC protein will be produced, and it controls the process of osmoregulation. As like the prokaryotic system, the eukaryotic system, that is plants and animals, show a very wide variety of signal transduction pathways depending upon the nature of the signal. And the general feature of the signal transduction is the signal interact with a receptor, and the activated receptor interact with a cellular machinery, producing a second signal that changes metabolic activity of target cell, and the transduction event terminated when cell returns to its pre-stimulus states. Four basic type of signaling pathway seen in the eukaryotic system. First one is the gated ion channel, and in this process of signaling, when a specific signal binds with the receptor of the ion channel, then the ion channel become open or closed basing upon the signals. The second type of signaling is the receptor is a enzyme complexes. When specific signal binds with the external site of this receptor, then enzyme activity will be takes place in the cytosolic sites, where particular enzymes will activate and cellular response will take place. Then third type of signaling process is the G protein mediated signaling. In this process, first signal binds with the serpentine receptor. Then that receptor activate the G protein, and the G protein interact or activate the effector enzymes, which produced a second signal, and that elicits the response inside the cell. And the fourth type of signaling is known as direct signaling. The signal directly enter into the cell and activated several receptors present in the cytoplasms or nucleus and binds with the DNA. Then that DNA expresses to form the protein and regulate the gene expressions. So let us start with signaling of steroid hormones. In this type of signaling pathway, the steroid hormones directly enter into the cell and it binds with a receptor present in the cytoplasm. It activated, the inactive receptor become activated, and the activated receptor along with the signals binding with the specific regions of this DNA, enhancer or silencer region of this DNA, and activate the gene expression. That means it transcribed to the mRNA, then mRNA to protein, and that protein mediated different responses inside the living system. Then signaling via Gproteincoupled receptor. And when water-soluble hormones binds with the cell surface receptor which activate the heteromeric G protein, the activated G protein activate the effective enzymes which generate the intracellular second messenger and which stimulates variety of responses. And this signaling process consisting of three essential components. One is called surface receptor protein, the second is heteromeric G protein, and the third is effector enzymes. So the receptor using heteromeric G protein is an integral membrane protein which are structurally similar but functionally diverse. The receptor protein consisting of 7 transmembrane alpha helix which have a outer or external domain for binding of signals and intracellular domain for binding of G proteins. This is called serpentine receptor because it moves across the membrane 7 times. Then the heteromeric G proteins, and the G protein that transduces the signal from serpentine receptor is called as heteromeric G protein, which located on the cytosolic phase of the plasma membrane. It consisting of 3 polypeptides, 1 alpha, 1 beta and 1 gamma. It always binds with the guanyl nucleotides, that's why it is known as G proteins. It cycles between active and inactive forms. In inactive states, the G protein is associated with the but when the ligands binds with the receptor, that induce the displacement of GDP to the GTP. And when the alpha subunit dissociates from this beta and gamma subunits, the alpha subunit containing GTP activate the effector enzymes, and that effector enzymes will produce the second messengers. Also, a GTPase activity, that is GTP is hydrolyzed to form GDP, and when it is hydrolyzed, the inactive alpha subunit inactivate the enzymes and alpha subunit with GDP reassociates with gamma and beta subunits. To discuss about signaling via cyclic AMP as the second messengers. And cyclic AMP is an important signaling molecule in both prokaryote and eukaryotic cells. Cyclic AMP is formed from ATP in the presence of enzyme adenyl cyclase, which is an integral membrane protein. When the hormones or signal binds with the serpentine receptor, then it activates the G protein by GDP replaced by the GTP in the alpha subunits. Then the alpha subunit containing GTP activate the effector enzymes, that is known as adenyl cyclase. And this adenine cyclase breaks the ATP molecule to form the second messenger, that is known as cyclic AMP. Then the function of cyclic AMP. The first function, cyclic AMP activate protein kinase A. All the effect of cyclic AMP in animal cell are mediated by the enzyme protein kinase A. In unstimulated cell, the protein kinase is inactive state, and in inactive protein kinase A consisting of 4 components, that is 2 regulatory components and 2 catalytic components. But when cyclic AMP attached with the regulatory subunits, then catalytic subunit become active, and that catalytic subunit phosphorylates the target proteins, and that target protein, it activate the inactive protein become active by the process of phosphorylations. And one of these example of protein kinase A mediated activation of glycogen phosphorylase kinase, which activate the glycogen phosphorylase, which break down the glycogen into glucose 1-phosphate in animal cell. Cyclic AMP also regulate gene expression. Cyclic AMP activates several transcriptional factors which binds with the promoter region or enhancer or silencer region, which regulate the gene expression. That's why in case of bacteria it is called hunger signal, because in the absence of glucose it can synthesize enzyme which explore other energy sources, such as lag operant system is activated by the cyclic AMP. That's why it is known as a hunger signal. And cyclic AMP also activate several ion channels. When cyclic AMP attached to these different types of ion channel, the ion channel become open or closed. Although there are some low level of cyclic AMP is detected in plant tissue, and the role of cyclic AMP in the plant signal transduction has been highly controversial, but some source of evidence support that cyclic AMP stimulate certain genes for pollen tube growth in plants. Cyclic AMP assimilates the potassium channel in the plasma membrane, and also it is believed that auxin mediated cell growth is also carried by cyclic AMP. This is all about the signal transduction process in plants and animals, and thank you for watching my YouTube video, and please don't forget to like, share and subscribe my YouTube channel. Thank you.